Welcome to Therapeutic Momentum. We take you on an inspiring journey, spanning breakthroughs in science and medicine development. Over the last century, our average life expectancy has increased by more than 20 years. Join us as we go behind the scenes with medical researchers, clinical developers, and other experts working together in the race to tackle life-altering diseases with new treatments and cures.
I'm Karen Akinsanya. I'm a translational scientist working at the intersection between biomedical research and emerging technology. Thanks for tuning in to Therapeutic Momentum. Let's get started. Well, it is my extreme pleasure to welcome my friend and mentor, Mathai Mammen, to Therapeutic Momentum. Mathai almost needs no introduction, but for those of you who don't know who he is currently the CEO of the newly minted Parabilis medicines. Mathai, welcome. Thank you for joining me.
My pleasure, Karen. It's a pleasure to be here.
I'm very interested in telling people more through this interview who Mathai Mammen is, some of the principles that drive you, some of the moments of inspiration that brought you to where you are today, really looking under the hood at some of the events that occurred along your journey to this point. So maybe we can begin with just a high-level overview of your career journey, and then we'll dive in and start to see some of those moments, pivots that really have formed who you are as a leader and experience as a drug developer.
Thank you. So the brief snapshot is I began my career really my professional training at Harvard here in town. I was part of the MD Ph.D. program, and it was through the Health Sciences and Technology division within the med school. And so that division offered me opportunity to take classes here at MIT, where we're near, and I grew the bug of, innovation and entrepreneurship during that component of the program. I finished my Ph.D. with George Whitesides in the chemistry department at Harvard. And from there, I had intent all along of being an academic, but potentially being involved in companies. And I'd seen models for that, including with George himself along the way. But instead, I did do a pivot at that point, and with the urging of George himself and others, ended up starting a company called Advanced Medicine and later renamed Theravance in South San Francisco in California. My wife and I, who had been dating at that point, got married and immediately moved to California to begin this company. And at Theravance, really proud, especially, and this is a theme that'll carry forward of two things. One is the people we hired and kind of launched into the ecosystem, and the other, the medicines that we were involved with and discovered. And after Theravance, after a long stint at Theravance, where I started my career as a scientist in the lab at the bench and grew up all the way to head chemistry, then biology, then research, then R&D, I moved to Merck, where we got to know one another, Karen, and where I was responsible for some of the areas of research. And then I moved from there to J&J, where I was responsible for all the research and all the development as the head of R&D for J&J's pharmaceutical division, Janssen. Although Janssen also, it's a time to rename, but Janssen was renamed J&J Innovative Medicine fairly recently. So as the R&D director of J&J Innovative Medicine. And then more recently, about a year and a half ago, I began a career back in the biotech ecosystem as chairman and CEO of Parabilis Medicines, newly named, as you just said.
Wonderful. So lots of different environments, lots of different experiences. Can I just go back to something you said, that you founded Theravance?
Yeah.
But you started in the lab. Can you expand on that?
Yeah, so that is a really interesting story. And, I'm not sure even in retrospect whether it was an error or a positive. But when I was thinking through whether or not to take my ideas into an academic career, so I'd begun the job application process and actually had traction at a couple pretty prominent places, or whether to take the road to create a company, how I even got the company idea into my mind is I was approached by Jim Tannenbaum. So at the time, Jim was at a venture capital firm, Sierra Ventures in California. So he was part of an MD MBA program. And he had spoken to George about new things that were happening. And George handed him my thesis. So he was the one that, was actually kind of amazing. He asked me to come to the Charles Hotel, which I had never been in because it was that period of life where you don't go to the Charles Hotel, you seek free food or pizza and beer. But he invited me there and he brought his own bottle of wine, which I never heard of anyone doing. But I didn't know anything about this. This was completely foreign to me to actually start a company like this. So I talked to lots of people. And one of the lessons I believed I learned at the time relates to the question you asked. I believe that there were actually a number of companies that had started out of Harvard and MIT and the Harvard teaching hospitals that I believed were good ideas. They should have worked out and had traction, but they didn't. And the reason I believed a bunch of the time was that these were young founders that were trying to hire seasoned people in an industry where experience was very important and could not. They could not. Mm-hmm. And I believed it wasn't the quality of the idea, but sometimes it was the arrogance of the founding team. So I said, if I'm going to take this 90-degree turn in life, I'm not going to make that mistake. And I'm not going to try to hire seasoned people, but rather do what I think I do well, which is the science. And these were my ideas that I wrote into the business plan. And I was going to lead or co-lead or make sure programs were led well, but not try to hire underneath me seasoned individuals. So I think I would have maybe done that differently today, but I entered as a scientist. And at first I was really happy because I was comfortable doing what I was doing, though that even was a stretch. I'd never been in a company. And I did hire, but more junior staff. And then I hired my boss, and then my boss's boss, and my boss's boss's boss. And at first it was fine, because I was a co-founder of the company. And even though I was in the lab, I was seen a little different. But as the years progressed, as we were in year 3 and structure started to be added to the company, I felt I had buried myself in my own company and I no longer had traction. So I wasn't invited to certain meetings and, it changes. As you know, there's a structure to a company for good reasons. So, but these good reasons didn't sit well with me. And I struggled, actually, to be honest. I've never really told anyone this story, but I really struggled with, did I make this awful error? What have I done? And I remember this time I was getting promoted, I thought, from scientist to senior scientist. And right when I was going to be promoted, the HR structure added research scientists between scientists and the senior scientists. So I thought, oh my God, this is nuts. But that said, in retrospect, I think about that because I did every job from really sometimes all-nighters in lab to supervising the various experiments that we did in chemistry, moved over to biology, was very intimately involved in all details of all experiments. Then when I came to be eventually head of research, I had a deep perspective on what was in the team's mind, what information transparency really looked like. Then when I eventually moved to development, and I was very involved in development at a pretty deep level, including protocol writing, but also presenting to FDA. I presented to FDA multiple times. And so these jobs that are through and through the company, I feel I've done them or I've been super close to all of them. And in a period in my life where I had plenty of energy and drive to, use natural curiosity to learn the ins and outs of all those jobs. So then when I was head of R&D, it called on the strategic mind and it called on other aspects that I learned as well. But I had the pleasure of being very close to almost all jobs in research and development. So by the time I got to Merck, I feel very calibrated on most every area of science. And Theravance cuts across all therapeutic areas. It was a platform company. So we had explored and had success in numerous areas, from infectious disease to respiratory medicine, to cardiovascular, to immunology. So we had been in all these different spaces. So I came out of that having had like a 14-year education across every area of research and development. So I regretted, like your question's a great one. It's an opportunity to really talk about that. It was born of a certain humility that turned into deep regret that now turns into gratitude. So it's interesting.
Absolutely fascinating. So one of the things that I hope to do is really get to these kinds of stories. What is it that makes Mathai Mammen such a great leader? I think you've just unlocked one of those in this story that you told about living the life of the scientist, founding a company and learning the job. So now then, as a leader, you have this understanding, deep understanding. You resonate with the journey of the scientist. I think it has a lot to do with the kind of leader that you are.
Thank you for prompting that. I don't think I've ever mentioned this to anyone in the last 15 years. It's so interesting.
One of the things that is very common, I think, scientists in general, but also drug discoverers, is the geographical journey that many of us have been on. You mentioned in Canada, you came to the US, to Boston, and then very quickly you were in San Francisco. Can you tell us more about the decisions to move like this, how they may or may not have impacted you, how that felt, not just for you, but also perhaps for your family, because these are not easy moves to make necessarily.
No, and it began even before the Canada portion. So I was born in India, in Vellore, in the Christian Medical College, which is actually an extremely well-known center. It's a medical school attached to a hospital, built around this mission of service. And there's a leprosy colony right next to the hospital, and everyone rotates through there. So my parents were technicians in a lab there. My aunt, my mother's sister, who's this absolutely fascinating, like she's my own personal hero, but she was an instructor, she was the head of the pathology department there and taught all the medical students. So we had a pretty good life there, actually. Like my parents, I was born there, my sister was born there after me. But my father saw all these people like rotating in and out of Vellore because it was famous for certain things, including the first kidney transplant using certain immunosuppressive regimens. People don't know this, but Vellore was one of the prime centers of figuring all that out. And so, interestingly, famous Judah Folkman, did a rotation there. My aunt went from Vellore to work for him at Harvard, which is kind of bizarre if you think about it. But that prompted my father to just get a bug in him to look all over, including in Europe, the United States, and Canada. But it was a lot harder than he anticipated to just go get a job. This was in the late 60s. He applied for hundreds of jobs. And it was a time where it was hard to even find information, right? There's no internet.
That's right.
So you wrote away for information. They may or may not write back. And then you send applications. They don't know who you are. Who are you?
Yeah, there's
You're
no
in some small
profile
town.
or LinkedIn or anything.
No, there's nothing. And so, but he finally got a job offer from one of the hospitals in Halifax, Nova Scotia. And he ended up moving there on his own at first. But you can imagine the culture shock. And I'm telling you this because as much as it'll appear that I made some interesting, good moves, I don't think I could do what he did because he came from the south of India to Halifax in January of 1969, where it was a snow-filled, it must have looked awful in some ways. It was a snow-filled environment that he walked to work from there. And he eventually befriended someone at his work that helped him buy a car and then taught him to drive the car. And so it was this like crazy time. And then so I moved with my sister and my mother after about 6 months to Halifax, Nova Scotia. We, of course, didn't know any differently, so we thought it was fine. And I didn't find that a culture shock, but I'm reflecting what my parents must have thought. Their English wasn't excellent, but it was good. And that still allows me to this day, by the way, to be highly forgiving on how people communicate and cultural differences. Especially with my mom, I feel like she was not well treated for her accent. And it's actually a very important part of who I am. But then one thing is they never really spoke English to me at home. So as I went to kindergarten, I was sat down by my father and said, you're going to school. They don't speak Malayalam in the school. They speak English. And we've spoken some English to you, but you'll have to figure this out. You'll have to figure out that this is not, that you're not going to be comfortable at first. Plus, we call you a name at home that's actually not your name. So your names from now on in school are going to be called Mathai. Oh. And so that was also, so the language and your name's not your name is your identity. So what do you mean? So I went to school and I literally had no idea what anyone was saying. I had no idea. And so it was this terrible experience, and that stays with me to this day. I had no understanding. By the end of that week, I was placed at the very lowest levels of these tiered systems that they had, because I said, this guy, we don't understand what he's saying. He doesn't understand what we're saying. And so I got put in these low levels. But eventually, by the end of that year, figured it out. That was a traumatic experience,
I can really imagine. I mean, I must say, I fortunately lived in a country where everybody spoke English.
Yes.
And so I ended up coming to America. Everybody speaks English of a form here.
Yes.
So never had to experience.
Yes, but yes.
Never had to experience that. But I must say, it can only have created a coping mechanism and a sort of, eventually at least, comfort with change, right? Because you'd been through this incredible change.
Yes.
And so I presume you've taken that now forward throughout your life and your career.
I don't mind being dropped into situations that are very unfamiliar, not just from then, but other times in my life as well. But I ended up growing up there, and we were a relatively poorly off family. We lived, at the time, again, as a kid, you don't know, but at the time I recognized would be the equivalent of the projects. This is very low income housing in Halifax. My parents were technicians, right? They were at the lowest level of their lab, and this was barely above minimum wage. But they really, we had a cozy little spot, and there were lots of kids around. So my sister and I had great fun, but we would spend vast amounts of time, just outside the house, totally on our own with other kids. So then eventually I grew more serious about a couple things, sports were really big, and then athletics were a very important part of my life. It was how to kind of escape the purely interactions about speaking and language early on, but then it became its own love. And math, interestingly, I was obsessed, from junior high through high school. I wasn't so much into science, but math, athletics, and languages. So languages became a big part of me. French, of course, was taught alongside English at all grades, from kindergarten on up, including to literature and spoken and written French. But then German and Latin I also really loved. And so I took those in addition to Malayalam and a little bit of Hindi also. So languages and the structure of them, and how, it's kind of an amazing thing that you and I can sit here right now and I can have thoughts that I formulate in this probably lossy kind of way. All those complex thoughts get formulated into words. You hear them, and I can influence your brain chemistry. So to me, language and communication like that are fascinating, that even works. Then that different people use different languages to structure that. I became a believer that depending on what language you spoke, you were capable of different kinds of thoughts. If you can put into words certain kinds, and English is so rich that it's almost unlimited. But there are ways of structuring English sentences that are different from German or Latin, different from Malayalam and Hindi and the Sanskrit languages in general, that make you think in a different order and might increase the kinds of thinking and analysis that you're capable of. So mixing people from different languages and cultures is almost always a good thing, because you get diversity of thought that's uncoupled from the education, but more about how they speak. And so I find that all very interesting anyway.
So sort of a linguist, I presume that language, love of math, actually your original story about living and learning every job, perhaps you can now tell us about how you think about bringing people together around a particular endeavor. Maybe you can talk a little bit about teams and maybe the sports that influenced you, even when there were struggles like language. Tell us more about how you think about teams. For those who are listening, I'm sure hearing how you think about teams will influence how perhaps they approach teams themselves.
Yeah, there's not like a way to think about teams. And even in my own journey with different teams in different environments, I continue to evolve. So there's no final answer. There's not a way that I think is the best way, but I'll tell you how I have thought about teams and how I've both succeeded and failed in thinking that way. But early in my career, it was very influenced by Roy Vagelos. So Roy was the original chairman. That's its own story, but how he was recruited. Like, I think he felt sorry for me at some level because I kept pestering him about this company that I was forming. And he engaged finally in a very serious way and agreed to be our chairman. But that said, like once Roy was there, 1997 onwards, I took every opportunity to learn from him and how he thought about teams. So he was very influential in my early thinking. He said that be relentless on finding the most talented people you can, worry a little bit, and this is now not how I think about things now, but worry a little bit less about how they might interact with one another and more about their capability to find a medicine or do their part. Like maybe they're a chemist or a biologist or whatever they are. Be deep and be almost a key opinion leader. Be so expert in what they do. But I remember the other part, but worry a little bit less about how they work with one another. Later in my time at Theravance, he had recently read a book called Team of Rivals. This is about Abraham Lincoln's time, who exemplified Roy's thinking in this space, where Abraham Lincoln had a set of people on his team that themselves were amazing, but each wanted Lincoln's job, and they had no love lost with one another. And so it was a team of rivals. And that net for Abraham Lincoln and presumably for Roy worked. Like you get these experts in a room, and Merck honestly was very much built that way, as you know.
Yeah.
And so you get these experts that are capable of extreme original thought and holding conviction on those thoughts and making good things happen. But it's sometimes not that fun, right? to work in a team environment where that's been the philosophy. So that was how I was early in maybe the first 5 years of my career. But I realized in time that that wasn't working for me. Maybe it would work for other leaders, but how important it was that there be the locking of puzzle pieces among the individuals on this team. How important that is. I realized, no one would say that you should give on anything. People would say get talented people that work together well. But usually in real life, as you build teams, there are puts and takes on different individuals. But right now, I recognize with great conviction that the right way is to optimize the whole. And some individuals are more apt to lead, some individuals more apt to follow. They're capable of great thought. There's different degrees of deference, though, that are very important in a team environment. And I now feel comfortable that that's my current style. That's my current style. I need this team to be way better than the individuals that comprise it. And if you're careful about that, you have to be also careful that the individual team members, like one of my first most important things outside of curiosity is humility. That if they come in with arrogance into that team environment, that plant seeds that will eventually turn that team bad. So you need team members that are simultaneously very curious and capable of new thoughts, and you need them to be humble so that they're capable of listening to others' thoughts and adopting those in preference to their own if that's the right thing to do for the team.
Right.
That's not an easy thing at all to do, at all. And but that's my current philosophy. And so if you look at the Parabilis team right now, it's an incredibly cohesive, awesome group that I'm proud to be part of. It's hand-picked and curated so to exemplify these features of high level of curiosity, first principles thinking, smart, and cohesive and capable of interlocking with one another in a way that very proud of. And it takes work, to make sure that the right people come into the company. It's not so much about the daily watering, though that's important, I'm sure, but it's more like make sure the right people come into an organization like that. Harder to do at a large organization where there can be 15,000 people on a team.
Right.
But easier to do when there's 120.
Yes. So we used to use the term intellectual sparring at Merck, right? Because people were really, experts in their area. But what I think you're saying is it's fine to have that intellectual debate and discussion, but the collaborative output of that has to be sort of raising the team, as it were, and helping that team reach new goals, new heights.
Yes, I think so. I think that the philosophy of Roy Vagelos can certainly be very, very successful. It has been. Like, who am I to say? He's been amazing.
Right.
One of the leaders of our industry over time and space, like an amazing thing. It's just not my own personal philosophy to be so extreme on that, be a key opinion leader, that you lose the ability to be humble and lock hands. Like, that's the piece. I won't trade that off.
I want to go back to the discussion about your decision not to hire very experienced and seasoned people and try and be their leader. You sort of set up this system where essentially you were junior and rising, but you were recruiting the leaders in your organization. A lot of people, I hope, listening are early career, and they're thinking about maybe they have new ideas, maybe they're trying to form a company. Maybe you can go back and talk about early career people having perhaps a seat at the table, allowing their voice to be heard.
Yes.
I think you and I both know, as early career scientists or drug developers, sometimes that's nerve-wracking to speak up in a room full of experts, full of people who've previously invented and launched drugs. I have a philosophy about allowing early career people to have a seat at my leadership table. I'd love to hear from you how you think about that.
Great question. I think my advice to the leaders of those teams where such early career people are invited are to remember that, the fresher the mind, the more free it is of bias, and it's more likely than not that a fresh new idea that hasn't been old and turned over again and again come forward. So that's a very important point. And it's one of this kind of bias that builds up in you, if you're not careful to dismantle it, that you can only counter with the fresh voices that are around you. And so no matter how experienced you are as a leader, one should remember, and this is again part of the humility, that you're biased. I got this, by the way, for the first time from George Whitesides, who invited us it was my first Christmas party at his house in Newton here in town. And so I walk into his house, and on the left side is a library where he works, or his office. And on the walls are stacked piles and piles and piles of journals still in their plastic wrap.
Okay.
And I was wondering, George, what's the deal with all the journals in their plastic wrap? Because Mathai is a really important point. It reminds me not to be biased by the literature. And so it's this phenomenal point. This is George Whitesides, who is one of the most amazing people I've ever encountered, and is more versed with the history of certain science and current thought, and is extremely well read, but he reminds himself not to be biased by the literature. So it's analogous to what we're talking about here, Karen, your question around young talent. That leader needs to remember not to be biased by their own experiences and allow for fresh thought. And then if I were in the shoes of the young career person, they should steel themselves and they should feel proud of their fresh thoughts, because they should know that the seasoned people are seasoned, yes, and should be listened to, but also have biases. So that should help with the conviction to have your voice heard. You can say, I know that all these people around me have these decades of experience, but I have an idea or I have an opinion, and you should feel confident speaking your mind, because no matter how seasoned, they've only lived a couple decades. It's not like they've lived 500 years. So they're limited. All human beings were limited. So they should feel confident about expressing their voices because they understand that the room is limited in that way.
So let's pick up that theme to talk about another interface, drug development as a discipline, now meeting tech.
Yep.
Two potentially very different worlds coming together, perhaps for the next leg of the journey of how we invent medicines. I'd love to get your perspective on this. I think when we first met at Merck, we were talking about the fact that there was all this incredible data that was locked away in ELNs and team sites and all over the place. And wouldn't it be interesting to think about all the knowledge that was locked up in those various places? But this was around 2015 or so. Almost 10 years ago now. Yeah. I think over the last 10 years, a lot has happened to bring tech and pharmaceutical research, biotech, really all together in terms of the next wave, if you like, of how we invent medicines, bringing the old and the new perhaps together in a little bit of a different way. What's your view of this and where are we headed?
Yeah, that's such a big question and has so many parts, but let me just take it in a specific direction. First of all, I think the conversation we had about a decade ago, both our instincts were correct. That there is a revolution coming, we should be part of it and not oppose it. But it's also interesting to me that you asked this question following the conversation on bias, because I do feel the first area that could be tackled just with unfancy, non-advanced algorithms, but just the existence of data is bias. So as an example from J&J, I think it's a good example that speaks to this power of low-hanging fruit of large volumes of data that can get opposed to bias is the recruitment into J&J's pulmonary arterial hypertension studies. So when the studies of the large phase 3s or the indications that were the phase 4s effectively after the first approvals were being constructed, it's a rare disease, pulmonary arterial hypertension. To recruit like 900 patients, which would be for that phase 3 trial that we wanted to do, it's like a 3 or 4 year long recruitment phase. It's a hard to find population. And furthermore, there were a handful, literally 5 key opinion leaders in the world that the organization wanted to be part of that study because they were the world's leaders, supposedly. But when you pick a person like that, as you know well in drug development, you get all the biases of who, their own sites that they're associated with, who's good, who's bad. They sit as judge of that. And we, at that point, my data science organization headed by Najat Khan, who's a phenomenal leader, had created systems for looking in the world at the various databases that would allow us to say where the patients were. And very interestingly, the where the patients were analysis was overlapping with, but quite different from where the key opinion leader said we should recruit.
Interesting.
So it's nothing fancy, right? It's just comparing analyses that came at it through key opinion leaders' lenses or totally unbiased data lens. And then I said, well, it's obvious then we should just go to where the patients are and our recruitment will go faster, because we had modeled recruitment rates and it was very dramatic how much we could cut them if we just go to where patients are and pick the sites there. The key opinion leaders said, well, those sites are no good because they're second-rate people that are in charge. They don't know how to do trials. Is that true? I remember asking, is that definitely true? So we did our own analyses of that, and sure enough, they were fine. They were hungry. They really wanted to. Yes, sometimes it took a little bit of bolstering from our ClinOps group, or talented ClinOps group, but it was far better, turned out. These key opinion leaders, there was risk that the teams perceived saying, well, they might just be out. They might not participate in our trial if we don't listen to them.
Yes.
But sure enough, we went with what the data science teams were telling us, and it was all fine. The key opinion leaders didn't go anywhere. They were not happy at first, but this unbiased approach that all that data brought to the table allowed us to recruit trials much faster. And that applies across all the trials that J&J was involved with. It's not a big change, right? And you would think it's obvious if you just get dropped into a system from outside, but it's actually a remarkably big change from within the organization that is accustomed to following expert opinion above all else. So to distinguish between expert opinion and unbiased analyses, which have their own potential bias, even though they're unbiased, maybe you've inappropriately weighted for expertise, or there's a different problem. But to be able to bring those worlds together created immense value when I was at Johnson & Johnson, immense value. Then if you start layering in all the stuff that you and I now spend time thinking about, like drug discovery and development, endpoints that are more mechanistically homogeneous, so you have the potential for precision in patient selection, drug discovery, generative AI right now is working really well on proteins. I'm involved in standing up a startup called Zera in California that we talk about, but there are all these things happening that are definitely harnessing advanced computation. But I don't want us to forget that first and foremost, there's a vast amount of information in science, in medicine, in clinical data that should be a stopping point for eliminating bias in both the creation of medicines, the conducting of clinical trials, and the practice of medicine. As you may be also following, in the practice of medicines, there's certainly biases in the system from socioeconomic, race, male, female, children, often their voices aren't heard. But all that data, objective data analysis, can create a system, if appropriately pushed and applied, that is free of bias in the asymptote, at least. So I would love to not forget that all that data has that potential before we start talking about advanced computation like generative AI, et cetera.
It's so interesting that you give that example. I think when we talk about drug development being this incredibly costly and lengthy process, the example you just gave is relatively straightforward way to disrupt perhaps the way we've been doing things. But if you add up many, many examples like the one you gave, you can only imagine that this process has become slow because we are almost sticking to the old way of doing things.
We have bias.
But at the same time, we don't want to necessarily just disrupt things for disruption's sake. And I think it's an interesting moment in time to think about where are we going to both test and validate how innovation, computation, removal of bias through more systematic looking at data. How is that? Perhaps not as headline worthy, but ways of slowly and incrementally optimizing this endeavor of drug discovery and drug development. I think that's what your example highlights.
It's piece by piece by piece, right? There's no way you can go into a complex organization that's set up and just say, we're going to eliminate bias, or we're going to introduce data in this way, or we're going to use advanced methods. So, maybe the J&J example is the best to illustrate this. But when I knew, like from our conversation, Karen, from a long time ago, I had great conviction that data science, broadly defined, is going to revolutionize how one does research and development and commercialization and manufacturing and most everything else. So that conviction was clear. But when you come into a big complex organization like J&J, I couldn't act on that conviction in every manifestation all at once.
Right.
Someone told me this analogy a long time ago that you can take a branch off a tree, and if you want that branch to go from straight to 90 degrees, you can get there, but you have to do it the right way. If you bend too quickly, that branch will snap.
Right.
If you bend at the right pace, you can get to 90 degrees. So pacing is everything.
Yes.
And so I took that to heart. And so I asked myself at J&J, what would be the mechanism to introduce concepts so that the branch wouldn't snap? but it would bend. And one of the things, and again, I thought these things, but they were executed by some amazing people, like that were on my team, including the leader, Najat, was excellent. But we introduced very pragmatic use cases in drug development. So the reason in drug development, not drug discovery, is the organization as a whole. For the organization, those that controlled purse strings, from the CFO, CEO, and all the budget holders across J&J, not just in R&D, the way they were going to recognize that this movement had value was to see examples that were here and now, that didn't take 10 years, but rather might take 1 year or 2 years. And so drug development was going to be a more fruitful place to do that than drug discovery initially.
Yes.
And so drug development, there were these incredible examples. So recruitment speed, use of real-world data as a contextualizing arm. Try not to say synthetic control arm too much, because I think that has connotations sometimes that are not good. But data that contextualizes single-arm studies, we use that repeatedly in our oncology approvals. So some of the approvals were very, very, very much supported by these arms that we put together very rigorously to know with certainty that what we were seeing in a single-arm study had great value for the patients relative to what they could have otherwise had as treatments. And FDA and EMA both really appreciated our advances methodologically in what we were doing. And eventually, by the way, gave J&J a grant to continue and help them write guidance on some of this. So these kinds of examples of clinical trial execution, of use of contextualizing control arms, they were here and now, and they drove the portfolio to higher value. We could get more done with fewer dollars, with less time. We were more apt to succeed than we would have otherwise, so drove up probability of success. It was very visible. Like, we always had a listing of these use cases that were assembled, and it changed the culture of the company. And then eventually Jim Swanson came in as the CIO and was pushing the same philosophy. And then together, I feel like the whole of the organization became a leader in data science as it pushed very aggressively on multiple kinds of dimensions and use cases. And then in my latter years was very much also in drug discovery. So Najat's effort were instrumental there. Tommaso, lots of other people that were very important. Emma Parmi, Rich Tillier, everyone leaned in the same kind of way. And great things started to happen on the key use cases there. And I hope it continues that same momentum right now. As I stepped away, I'm obviously not involved in that anymore, but there's sort of a story of how to drive change in large complex systems that thematically begins with the choice of problem that is most apt to be solved in a short time frame, that is objectively valuable, that in some ways is more important than choosing a problem that may even have higher value but takes much longer and is going to be less obvious. So this is the arc of change and how to drive it.
I think we as an industry have made tremendous progress. We have wonderful drugs that are really helping patients now. But we still have a long way to go. There are many diseases where there are absolutely no treatments. There is no playbook for even how to develop medicines in some of those areas. As you think about your past career and you think about the future, maybe you can do two things. One, what are you thinking about the future and going after those existing unmet medical needs, patients who do not have treatments? And maybe as you tell us about that, maybe you can tell us more about what inspires you to continue this journey of inventing, designing, developing, and getting drugs approved.
So where do I see some of the developments in the future, where the biggest needs of change? I do continue to see like the revolution that's happening in biology right now is continuing. There is another revolution in molecular sciences. Part of what Parabilis does is try to drive that with a new modality with alpha helical peptides. There are revolutions also in the construction of those through living therapies like cells and viruses. And then all of that is in parallel now to revolution in technology and data science. And that includes the systems by which data is collected and cleaned, the various both predictive and generative algorithms that sit on top of that, as well as advances in simulation, like molecular dynamics, like what Schrödinger has led the field in. Like these are all the advances in technology. So in the sciences and in technology, these revolutions are happening. Sometimes they marry up, but often they're just happening on their own. So I see from what fuels change as the marriage of those two parallel revolutions. Then on the parts of medicine that need to change, I'll start with the thing that most bothers me. Actually, it really bothers me, is that our medicines they often don't have nearly the impact we hoped when we started working on them, or even once they got approved.
Right.
They're effective in a very variable sense. Like, the drug Stelara, J&J's amazing drug that's used right now for multiple indications. If given to a patient, say, with Crohn's disease, for some of those patients, it's of immeasurable impact. It changes their lives completely, but for most of those patients, does not. And for a portion, it doesn't do anything at all. But yet, the patients use those medicines and they eventually cycle off and they go on something else. But it's the minority of patients for most of our medicines, even the drugs that we consider really high impact, great medicines. It's the minority of those that use them that actually derive the most impact. And to those where it's not impactful, they still suffer the side effects. So this kind of imprecise, so to speak, use of our medicines and the insufficiency of our medicines in general, those two facts of life really drive me. The precision element right now has a term, precision medicine, but precision medicine is not really a thing outside of some very small areas. So, if you have gene replacement therapy for a genetically diagnosed condition, but that's only a small part of medicine. You have oncology that's moved significantly in that direction. And right now there's such a thing as a precision oncology, a targeted therapy. Beginning with Gleevec, it's really opened a field. And now we're more motivated to try to find response signatures and use of DNA and transcripts right now, both, maybe in the future images. But cancer and precision cancer is also like a relatively narrow part of all medicine.
Yes.
And then viral disease, interestingly, is a third example where medicines are used precisely. You're said to have HIV. You're not said to have Kaposi sarcoma. Your disease name in that case is the underlying root cause of your disease and not a clinical manifestation. Outside of those three areas, so rare genetic disorders, certain forms of cancer, and viral disease, the other 90% of all medicine is phenomenological in many ways. These diseases, including, I shall contrast, bacterial disease and viral disease. Shocking. They're both infectious diseases.
Right.
They're departments within the same companies, right? Infectious diseases. But one is precise and one is not precise.
That's right.
So we make drugs for pneumonia, bacterial pneumonia, and we try to find, broad spectrum gram-negative, broad spectrum gram-positive agents. And drugs are in fact put to one side that are too precise for one organism. And, Merck had an absolutely gorgeous program in LPXC. It was a long time ago.
That's right.
But it was a beautiful program and eventually got put to one side because they couldn't get activity against Pseudomonas aeruginosa, but they had amazing activity against E. coli and some other gram-negatives. So precision medicine in that case was possible, but discarded.
Right.
Shockingly.
Yes.
And then these clinical diagnoses, some from descriptions using our senses, or maybe some famous person in the past, Alzheimer's disease, Parkinson's disease. These are named by syndromes, that your set of information's collected using what we can measure, what we can see, what we can hear. and diseases are named that way. The very axes that are used to do those namings, almost by definition, will yield heterogeneous conditions to be clumped together, and some conditions that should be part of the same disease to be called separate diseases. So this is a complete mess, in a sense, outside those three areas I mentioned. And my dream would be to use all this technology to slowly make headway towards actual precision medicine. But it's a daunting task,
Yes.
because it's not just going to be done and dusted by the scientists that recognize it, because there's an entire medical practice. Like, I use another example just from my past with IL-23 blockade. So the indications for Stelara and now Tremfya are in psoriasis, psoriatic arthritis, ulcerative colitis, and Crohn's. Those are the big four indications for Stelara, say. But if the condition is driven by, if those conditions are driven by overactivity or too much flux through an IL-23 pathway, and they could be alleviated by that, there's something clearly in common about those four conditions. And what is that? And could we identify that? Could we call whatever that commonality is the disease? And that disease is homogeneous mechanistically and would benefit all those patients with that mechanistic disease, regardless of the clinical manifestation, should be treated with this great medicine. You would drive response rates to 100%, again, in the extreme, because all of them would be homogeneous and responsive. But we don't think of medicine that way. And then the same is true for other conditions of the autoimmune type. Like, can we put boundaries around them? Can we draw with a pen on the right molecular signatures, and then they don't have to be DNA and RNA, it could be other kinds of signatures. Could we draw boundaries around those that are mechanistically homogeneous? That's the name of the game. And if they're mechanistically homogeneous, they should respond homogeneously to a mechanism.
Right, exactly. This is where potentially technology and the ability to accelerate experimentation, longitudinal data collection should begin to, one hopes, surface some of this sort of thematic biology that we can then address with precision medicine.
Yes.
That's the hope, anyway.
It's happening in certain companies. Like, I tried to do as much of that as possible within my older job at J&J. Like, for example, within psychiatry. Psychiatry's been a phenomenology for a long time.
Indeed.
And so depression, as an example, we drove pretty hard the use of esketamine, Spravato, with certain kinds of patients defined in certain ways. There's two additional programs. One is the blockade of orexin, and one is a kappa opioid agonist. Both of these, like say the kappa opioid one, those patients, this is Bill Martin and team, have very nicely defined a population that looks at a machine learning algorithm across psychiatry questionnaires, not molecular data right now, but questionnaire data, and find that the response signature to that molecule, to that kind of agonist, is a patient with high levels of anhedonia. So not just generalized depression, but that particular feature seems to stand out. At least that's the English term given to what is ultimately a pattern in a machine learning sense. But it seems to be anhedonia. And Wayne Drevitz, Bill Martin, and that team, Hussein Imanji before, did a lot of work to try to understand that biologically, and that the proof will be in the pudding. The phase 3s are ongoing, but it's a precision-driven psychiatry program. And orexin, it was the same kind of thing on an insomnia index scale. So these are examples where at least the industry, the pharmaceutical industry, is attempting to take something that was very vague and make for a signature. In the kappa agonist sense, it was in retrospect attempted to be understood biologically, like why that would be responsive. But I'm saying, like, even if it's not understood that way, that's the response signature.
Right.
And so as a first step, let's use what the data are telling us. Let's organize our patients in the way the data are telling us. You talk to Rupert Vesey. Rupert would say correctly that these precision signatures, sure, in an ideal way, they be there and we should use them. But so far, he has not seen examples of sufficiently sharp boundaries on precision signatures. So meaning a biomarker positive group might be 30% more likely to respond. So it's not black and white.
No.
So we have to do a better job on these signatures to get to more black and white, categories so that we can better select the patients that deserve our medicines. All medicines that are prescription medicines always have some downside, like whether adverse effects or something, right? That's a downside. And they're less apt to have those if you precisely choose your population.
And schizophrenia is obviously a disease where I think those patients have suffered a lot. I think
No question.
If you have diabetes or a heart attack, people have a lot of empathy for these kinds of diseases. But diseases of the brain in particular, I actually, one of the first phase 1 studies that I did at Merck, I was working on a schizophrenia drug and we were imaging the brains of these patients. And it really struck me just how little we could offer these patients. So I'm really excited to see a return to trying to understand how to develop safe, well-tolerated drugs for schizophrenic patients.
I do think, by the way, that you asked about like future predictions. There are certain areas that are very large. Like if you look at the WHO for the diseases that cause big morbidity, mortality. From a mortality standpoint, they're mostly diseases that are causing ischemic events to the brain and heart.
Mm-hmm.
So heart attacks and strokes.
Yes.
Morbidity is a huge amount of psychiatric illness. Huge. And so these areas right now to tackle with precision, to me, would be the biggest impact on kind of the area under the curve of the health of humanity. Those would be the areas. So, for example, the up-and-coming Factor 11 programs in the cardiovascular space, so Milvaccine, the collaboration between J&J and BMS, and there are others right now in the mix. The biggest reason people don't use Xarelto and Eliquis, the 10As, is bleeding. So only a very small fraction of patients that should rightly be on that, where the risk-benefit makes sense, use those medicines for fear of bleeding. So my question is, can you use data science right now to identify who's most at bleeding risk? Because not everyone is.
Right. Exactly.
And amazingly, if you look at the guidelines, and the guidelines tell you like who's at high bleeding risk, there are features described in the big cardiovascular guidelines. But if you double click and you said, well, why is that? What's the strength of evidence? Very poor. So this notion of precision can be applied to these vast areas and either emphasize the efficacy, like these are the patients that we should drive, or in my bleeding risk example, emphasize the safety. These are the patients that shouldn't use this, but everyone else should feel pretty good.
Right.
And you would then drive those kinds of medicines towards patients that should rightly be using them and have nothing to worry about and are going to be highly beneficial in preventing strokes and myocardial infarctions, but they don't use it out of fear because they don't have guidance like this. So I'm very passionate about this. Like there's so much to do in this precision space.
And it's so difficult to come up with a medicine. I mean, imagine the years and energy and fundamental understanding of the genetics of the factors family. It took a long time to get to those medicines. They work, but how do we ensure that they get into the right hands, treat the right patients, so that all of that investment isn't lost?
Yes.
So, Mathai, we're coming close to the end here. Obviously, you've had a tremendous career. I think there are probably moments and memories along the way that continued to inspire you, because this is a hard job. We are one of those industries that face more failure, as we've just talked about, than success. What are the sort of memories that stick with you that essentially inspire you to continue working in this very challenging space? Anything you can share with us?
Yeah, so there are various memories, and then there's one major memory. But there on the people development side and on eliminating bias around how one treats people. I do want to just talk about my mother again on this. Like I watched her. So she was a technician in a cancer lab and then a neuroscience lab at Dalhousie University, where I went to college in Halifax. And she's an awfully capable person. My mom is, and she is every bit as capable, I think, as some of the better scientists in our industry. But she had a bachelor's degree and she was a technician, and her language was not perfect. And I watched how people treated her. And it was part for the language. It was in part because she was a woman in a time where this was in the early 70s and through the 70s and 80s. It was terrible, Karen. And so it left a long-lasting impact on me. Her older sister was my aunt that I described, that was the head of pathology at Velour. So this is an unbelievably capable family. And to watch her be mistreated like that was terrible for me and left an indelible opinion on all people are equal. All people look past like the small things and get to the big things. Like, what's the nature of their ideas? What's their willingness to execute and really commit to seeing those things through? What's their ability to work together? Like, these are the things that matter. The specifics of language and culture and, someone's religion and someone's any choice they make in life, these don't matter. So that's like an extremely important feature of my life. And it was really my mother's experience early on that influenced that. And I would say that's true for my father too, though my father somehow figured it out and ended up running all the labs at IWK Children's Hospital, where he started as a technician. But these are things that this how we treat people are a big piece. There were other things that happened that way along my life. Scientifically, there were many things where I was wrong about, like we had a 5-HT4 agonist at Theravance that I developed for GI conditions. So we were developing it for constipative conditions. But, in the end, there was a small QT prolongation, and those are huge lessons learned because that was like 8+ years of hard work that ended poorly, and us fighting FDA around that and risk benefit. So it was that lesson was go after the conditions that really matter, go after conditions that are leading to significant morbidity and mortality, that are grievous, as our old boss, Roger Perlmutter, used to say.
Yes.
But that's right. There's so much of that. So it's another lesson. And, there are all these downtimes that we have, and I get frustrated too. But I remember the very sick people that are actually out there and dying. I don't know if your listeners will be aware, but I had 2 tragedies in my own life. And the loss of our daughter, Mia, who was 6 when she died, that, to this day doesn't have a diagnosis. And that bothers me every single day, that there are people that are sick and that die, and we have no idea why. Like, we usually filter out all those kinds of diseases and conditions when we talk within the pharmaceutical industry. They don't even come up for discussion.
Indeed.
They're just unknown.
Yeah.
But if you look in the hospital, if you look in children's hospitals right now, there's a whole bunch of patients that have unknown conditions or that just get sick, and we don't know why. And so there's much, much more unknown than known. That's very motivating to me. And that Mia died I can't even believe it in some ways, because like, we grew up in educated settings and we have access to everything, but I could not for the life of me figure out how to help her. And it's something I carry a lot. Like, it's incredibly hard. And then our daughter, Lainey, passed away. And this is something that I think is so unfair. Like, she was born incredibly healthy, and she and my son, Matthew, very precocious, very bright, empathetic, like beautiful children. And Lainey had an appendicitis that turns out in retrospect that was missed on going to Stanford. So this was Lucile Packard's Children's Hospital's like top emergency room. And another clinic that we went to as well because we were concerned. And it was missed, and she tragically died. And oh my God, Karen, like, I felt like my wife and we would not be able to even go on. Like, we stopped living for a while. We had the love and support of a large community around us, including at Theravance, which I'll be forever grateful to, like those colleagues there. And that every single day I think about Mia and Lainey. They're very much stitched into my heart, and even though if I don't talk about them every day, like we're talking about now, they're in my mind constantly. And they're the reason I get up in the morning and I work hard to try to make medicines. They're very much the center of my life. And I can't even begin to describe like all the emotion around that. Yeah. But in terms of inspiration, your listeners should know that there are many Mias and Lanies and parents that are sick. And the work that they do every day that might help those patients also help the people around, like the people around Mia. Unbelievable, Karen. Like there were so many people around her that loved her and cared about her and were distressed on bad days and were happier on good days. So a medicine that would have helped her, or a medical procedure, but a medicine in our case, would have not only helped her as an individual, but would have had this ripple effect on everyone around her, her family, her caregivers. So the impact that our community of people that you and I are privileged to work with, the impact they can have on the world in terms of health, yes, but in terms of emotion and well-being for the people directly and indirectly that our medicines help, it's so profound. It's like one of those things like you can't envision very large numbers. It's the same kind of thing. Yeah. That the impact we're capable of is so profound that we can't actually grasp it. But know that it's there and know there's no better industry in the world to work in than the one we do because of that fact.
That is so beautifully said, and it must have been an incredibly difficult time, but that stays with you. I don't know that I've ever told you this, but I was a teenager growing up with a very sick parent. That continues to inspire me to, as you said, try and solve even one of these cases that are unexplained, to be able to contribute to the suffering that is going on worldwide. Many more cases, unfortunately, like your family had to go through, whether you're a child of a sick parent or a parent of a sick child. I think the industry in the public eye gets a pretty bad reputation in general, but there are people like you who are really trying to move the needle for all the patients around the world. And I think it's important to remind perhaps people not working in the industry that we're privileged to work in, that is what gets us up every day.
It really is.
Thank you so much for sharing all of these wonderful stories, all of these wonderful memories that inspire you to continue doing what you do. I think for those listening, there are so many lessons about what it means to be a great leader, what it means to be inclusive. I really appreciate you spending time with me. Thank you.
Thanks for the opportunity, Karen.